Hi, my name's Kylie Savage. I do biology A-level. And in this video, we're going to be looking at biotechnology and looking at the standard growth curve. Now, start off with, what is biotechnology? Well, it's the use of living organisms, such as bacteria and fungi, to produce food, drugs, and other products, such as the hormone insulin. If you want some examples, yeast, which is, I am going to apologise for my horrendous pronunciation, Saccharomyces cerevisiae. We'll go with that. That makes bread, wine, beer. Bacteria such as lactobacillus is lactose sugar, which produces lactic acid, which denatures milk, protein to yogurt and cheese. Corn, Fusarium viniferarum, and insulin, which is produced from genetically modified E. coli. So, right, sorry, I just completely forgot what I was saying then. Yeah, biotechnology. What's the use of microorganism biotechnology? Well, many biotechnological processes make use of microorganisms, and the use of bacteria and fungi in various processes is widespread for many reasons. For example, microorganisms grow really rapidly in favourable conditions, with a generation time of as little as 30 minutes. Generation time is time taking for numbers to double. They can also produce proteins or chemicals that are given out into the surrounding medium and can be harvested. They can be genetically engineered to produce specific products. They grow well at relatively low temperatures, much lower than those required in chemical engineering of similar processes. They can be grown anywhere in the world, which means that they do not depend on climate, so they can be produced at any time. They also tend to generate products that are in a more pure form than those generated by chemical processes. They don't need to be purified. And they can often be grown using nutrient materials that would otherwise be useless or even toxic to humans. And what are the growth requirements for biotechnology? Well, they have to have many things, such as the temperature has to be perfect. If it's too hot, the enzymes will be denatured, and too too cool, and growth will be slowed. They need pH, constant pH, as changes in the pH could lead to enzymes being denatured and reduce the activity of enzyme and reduce growth rates. Oxygen concentration. Most commercial applications use the growth of organisms under aerobic conditions, so sufficient oxygen must be made available. We also need the time and the addition of nutrients, due to the fact that the growth of the more microorganisms require a nutrient supply, including source of carbon, nitrogen, and any essential vitamins and minerals. The timings of the nutrient addition can be manipulated depending on whether the process is designed to produce a primary or secondary metabolite. But what is a primary and secondary metabolite? Well, primary metabolites are substances produced by an organism as part of its natural growth. These like amino acids, proteins, enzymes, nucleic acids, and the production of primary metabolites matches the growth and population of the organism. So we've done primary metabolites, but what are secondary metabolites? Well, these are substances produced by an organism that aren't part of the normal growth. The antibiotic chemicals produced by a number of microorganisms are almost all secondary metabolites. And the production of secondary metabolites usually begins after the main growth period of the organisms, and so doesn't match the growth population of the organisms. So now we're going to look at the growth curve for bacteria in a closed system. And it looks something like this. I will show you it in the official one in the book, but it comes out weird and it's all hard. So I've just done like a little diagram in my book. You can tell how awful my handwriting is. So here we go. We start off with the lag phase at the beginning. Here. Now the lag phase is when the organisms are adjusting to the surrounding conditions, and this is really, really slow reproduction. The cells are active but not reproducing, so the population remains fairly constant. Next we get the log or the exponential phase, the little line going all the way up here. This is where the population size doubles. There are loads of nutrients, so they can divide at a maximum rate. In some bacteria, for example, the population can double every 20-30 minutes in these conditions. The length of this phase depends on how quickly organisms reproduce and take up the available nutrients and space. Next, sedationary phase, this one right here. This is where the nutrient levels decrease and waste products like carbon dioxide and other metabolites build up. The rate of growth slows down, like what I said. There's a lack of nutrients, and the organisms are born at the same rate as they're dying, which is why it is stationary. In an open system, we're looking at an enclosed system, but in an open system, this will be the carrying capacity of the environment. Finally, we get the very sad death phase, or the decline phase. This is where the rate of growth slows because of the lack of nutrients and accumulation of waste products. Basically, nutrient exhaustion, increased levels of toxic waste products and metabolites lead to the death rate increasing above the reproduction rate, and eventually all organisms will die in a closed system. So I think that's what I was going to go through in this video. Was there anything else? Oh, also, examiners often ask questions relating to this growth curve. So you shouldn't be able to describe only what it looks like, but also what happens at each stage. And also how changes like adding nutrients or removing waste products would affect the shape of the curve. We can also go through the difference between batch and continuous cultures. A batch culture is where microorganisms' starter population is mixed with a very specific quantity of nutrient solution, and then they're allowed to grow for a fixed period with no further nutrients being added. At the end of each period, the products are removed and the fermentation tank is emptied. Penicillin is produced using a batch culture of penicillium fungus. Then, a continuous culture is where nutrients are added to the fermentation tank, and products are removed at regular intervals or continuously. Human hormones such as insulin, they're produced from continuous culture of genetically modified E. coli bacteria. But what are some advantages and disadvantages of both? Well, in a batch culture, the growth rate is slower because the nutrient levels decline with time. But in the continuous culture, the growth rate is higher because they're continuously added to the fermentation tank, so there's no chance for it to decline. But the batch culture is a lot easier to set up. If you think about it, at the end of each time, the products are removed and it's emptied and all cleaned out. So it doesn't need to be set up as specifically as a continuous culture, because in a continuous culture, the required growing conditions have to be continuously maintained. Any change in it, any change in temperature or pH or anything will ruin the batch. In batch culture, if contamination occurs, only one batch is lost, so that's fine because it's separate every time. But in a continuous culture, if contamination occurs, huge volumes of product may be lost. However, continuous culture is way more efficient, seeing as the fermenter operates continuously. But in a batch culture, it's less efficient as the fermenter is not operational all the time. So, by the way, if we're talking about contamination, that's bad because unwanted microorganisms do stuff like they reduce the yield of useful products from the culture microorganisms. They could compete with the culture microorganisms for nutrients and space. They could cause spoilage of the products and produce toxic chemicals, which could lead to the destruction of the culture microorganism and their products. So asepsis is vital in biological processes, and the term aseptic technique refers to the measures taken to ensure asepsis. That is, the contamination of the culture does not occur at any point from isolation of the initial culture through scaling up, fermentation, and product harvesting. So that is basically loads of stuff about biotechnology, and basically all thrown together in one. That was spread, if you're looking in the textbook, that was spread over 3 pages, just taking notes from each. So good luck in your AT biology exam. I hope that made sense. Bye.